Hello, everybody. This is the slide that goes with the activity for prokaryotic gene control, and we are going to make the trip operon. Your finished product will look like this. The materials that you will need is the template, and then one strip with the cutouts. There are 8 on one paper, so that means one paper is good enough for 2 tables. You will need several different colored pencils, a glue stick, a normal pencil or pen, and scissors. The first step is you cut off one of the strips from the paper with the 8 cutout strips, and you can use any colors that you want to color in the different shapes. Write RNA pol into these 2 shapes. That's the RNA polymerase. And then you will cut this one. You will cut the shapes out. Most of them you don't have to cut out to shape, so that saves you some extra time and work. Like this one here, just they stay together, just cut it out like this one, and this one. And then the 2 RNA polymerase, cut them kind of to shape. And this one, only the top margin needs to be cut to shape so it can fit into something else. So here's your template now. And we're going to write the title up on the line on top, and that's trip operon, so TRP operon. Then we're going to start labeling it. So this is a DNA strand, and we label this with DNA. The 3' end is on the left side, 5' end on the right side. This whole sequence here, from here to here, this is the trip operon, and we label that as trip operon. It contains the genes of the operon, so those are the genes that are being controlled. Those are the 5 boxes here, and we just write genes of the operon on top. And then these 2 boxes here, that's the promoter. The names of the genes are trip E on the left side, trip D, trip C, trip B, and trip A. So we have 5 different genes that are actually making only 3 enzymes. This with the funny shape here, this is the operator. Operator is part of the promoter. And then we have another gene. This is the regulatory gene, which we label here and on here as trip R. And this gene also has a promoter. This promoter does not have an operator, so it's smaller and doesn't have the second box. Next, we're going to start filling in some colors. So we use the same color for the 2 promoters, the one in front of the regulatory gene and the other one in front of the operon. And use a different color for the genes. Another color for the operator. And then the rest is just DNA. So I drew this as a helix here with yet another color. So after the genes of the operon, we have just DNA, but between the regulatory gene and the operon, there's also a stretch of DNA. So they're not right next to each other. And then, of course, on the left side of the regulatory gene is also more DNA. Now we can also take one of the RNA polymerase cutouts and put it right here. So it binds to the promoter, which then enables it to start transcription of those genes. So here we write transcription and we draw the mRNA strip here. And now, since these are 5 genes, each of them has a start codon and a stop codon. So we draw the start and stop codon in start codons as green lines and stop as red lines. It's a little bit hard to see on this. So the first line is green, that's your start codon. And then the second line is red, that's the stop codon for the trip E. Right after that, another green line. And then the red line for the stop codon for trip D, green line for the start codon of trip C, red line for the stop codon, green line for the start codon, again, red line, green line, and then the last red line is the last stop codon for trip A. And then you can color it any color that you want. Next step is translation. And like I mentioned earlier, the 5 genes encode for 5 polypeptide chains, but 2 of those enzymes that are made have a quaternary structure. So that means A and B together make one enzyme, C makes one enzyme, and then D, E together make another enzyme, and we label this as the enzymes. These enzymes, of course, make tryptophan, which is important for the bacterial cell to survive, and it can make tryptophan itself. Here's the precursor, so some material, some chemical that is worked on first by one enzyme, then by the next enzyme, then by the last enzyme, and then has the finished product, which is the tryptophan. So we glue this one here and write product tryptophan. At the same time, we also transcribe and translate the regulatory gene. So here's the transcription of the regulatory gene. It's a small mRNA and goes on to translation and makes this product here. So you can glue this here. This is the repressor, which is a protein, and is currently inactive. It does not have a shape that would fit into the operator. So that means it can't bind to it and therefore is inactive. Now, if the cell encounters a lot of tryptophan in the environment, then tryptophan on the inside of the cell will accumulate because the production is still running. This is by default on and constantly makes tryptophan. And then as a result, the tryptophan will accumulate. So we write down here, tryptophan accumulates. And then we do the same coloring in of the DNA strand and the labeling, just like we have it up here. So whether tryptophan accumulates or not, the transcription and translation of the repressor still goes on. So here's transcription, here is your mRNA band, and then translation. Once you got that, you take the next picture to glue in, which is this one here. So the repressor is made, and because of the tryptophan, the repressor and the tryptophan will now run into each other. The tryptophan will bind to the repressor. Therefore, the tryptophan acts as a co-repressor. So it binds to the inactive repressor, which causes the repressor to change shape. And with the shape change, it becomes an active repressor. This active repressor now moves to the operator and binds to it. And now, because it's active, it will also fit. So you glue this one right here. And as a result, the RNA polymerase cannot bind, cannot attach to the DNA, and cannot make transcription. So the result is RNA polymerase cannot attach, and no transcription of genes is happening. And that reduces the number of tryptophan in the cell. And at some point in time, if tryptophan is needed again, this molecule will detach, which causes the repressor to detach from the operator. The operator is now open again. RNA polymerase can attach again and can transcribe the enzymes to make the tryptophan. So this is the whole product. This is what it looks like when it's completed. And some important things to remember about prokaryotic gene control. The trip operon is switched on by default. It is part of the negative gene regulation because it can be on or off, and it is a repressible operon. That means it is always on, but it can be repressed when the products are not needed. Thanks for watching.